Hello, am I audible?
Yes, ma'am.
Okay, we'll wait for some time to let others join and then we'll start the session. Hello everyone. I hope all of you can see my screen.
Yes, ma'am.
Okay, so welcome to the 11th day session for Drug Delivery Principles and Engineering. And these were the topics that were covered in the previous week: gene delivery and the related vectors, polymers, as well as genes as vaccines, and vaccines using gene delivery and other variants, cancer vaccines, and immunotherapy. And based on these topics, I have some previous year assignment and some more questions which we'll solve today. Okay, starting with the first question. Here you have to select the correct option with regard to vaccines for cancer. Which one of these options you think is the correct option?
Option 3.
Okay.
Option 3.
Okay. Yes. All of you are correct. It would be option 3. By isolating cells from autologous tumor tissue, a wide range of antigens can be presented. So you isolate the cells and then lyse and inject with adjuvants. This, along with targeting the entire spectrum of antigens, it will enhance the immune response against this. But there is also risk of autoimmunity, and it would need a lot of tumor sample, so it requires surgery as well. Okay, we have the next question. You have to fill in the blanks with the correct option. So, therapeutic cancer vaccines are a type of active dash with the aim to produce dash immune response to tumor antigens or tumor associated antigens that are able to dash cancer cells.
Option 2.
Option 2. Okay.
Option 2.
Okay. Anybody else would like to answer? Okay. The right answer is option 2. So, here the therapeutic cancer vaccines are a type of active immunotherapy with the aim to produce a specific immune response to tumor antigens or tumor associated antigens that are able to destroy the cancer cells. Moving on to the next question. Dash polymers are generally used in gene delivery.
Cationic and positively charged.
Okay.
Option 1 and option 4.
Okay. Anybody else would like to answer? Okay. Both. Okay. So both of you are correct. It would be cationic and positively charged polymers. So DNA is for gene delivery, you either use DNA or RNA, and both of these genetic materials are basically negatively charged. So you would want something positively charged or cationic to interact with this so they can form complexes, and it's easy to modify them to attach to this kind of polymers. Moving on to the next question. What makes chitosan a suitable polymer as a gene carrier?
Option 1 and 2.
Okay.
Negatively charged.
Sorry. Can you repeat again? Anyone else would like to answer? What makes chitosan a suitable polymer as a gene carrier?
Option first and second, madam.
Okay. Yes, both of you are correct. It would be positively charged and biocompatible. So these chitosan have amine groups attached that provides this positive charge onto this polymer, and this basically can interact with the negative charge DNA. Also, it is naturally derived, so it is biocompatible as well. Natural polysaccharide. Okay, moving on to the next question. Which of the following polymers are not commonly used for gene delivery?
PBC.
Okay,
PBC.
okay,
PPC.
yes, all of you are correct. It would be PVC. PVC is a common polymer that is used for pipes, wire, cable insulation, plastic bottles, and other things. Apart from that, all of the other polymers, polylysine, polyarginine, polyethylenimine, and chitosan, are commonly used for gene delivery. Okay, we have the next question. Which of the following is false for allogenic tumor cell vaccines?
Option 3.
Okay,
Highly successful approach.
okay,
Option 3.
okay. Yes, all of you are correct. It would be option 3. It is not a highly successful approach, and all of the rest of the options are true. Basically, use of very few established tumor cell lines. There is limitless, so it is a limited source and cost effective, and there is the risk of autoimmunity, and it may not be effective at all. Okay, moving on to the next question. Which of these is not a potential tumor immune evasion mechanism?
Last option.
Okay,
Last option.
okay. What about others? Anybody else would like to answer?
Down regulation of MS.
Okay, Anyone else? Okay, all of you are correct. It would be upregulation of antigen processing. So, most common potential tumor immune evasion mechanisms include physical barriers imposed by the tumor architecture. It's very complex, so it is difficult to for immune cells to interact with the tumor, uh, with the inner cancer cells, or the complexity of the structure that the cancer cells provide. And then induction of host myeloid and lymphoid suppressor cells, blocking of T cell effector function. So these tumor are able to suppress the host immune system. Further, it evades the T cell recognition, but it does not upregulate antigen processing. Okay, we have the next question here. What are not the objectives of gene therapy? You have to select two correct options here.
Last two options.
Okay.
Third or fourth option is correct.
Option 5 and 6.
Okay.
Option third and fourth.
Option 4 and 6, madam.
Okay.
Option third and fourth.
Third and fourth. Okay. Okay. So the correct answer would be the last two options. Cleave healthy genes already present to generate haploid cells and cause dysfunction at a cellular level. Here it is asking which of these is not objectives. If you look at the third option, it's about produce therapeutic proteins that help treat a disease. So you're basically treating the disease with therapeutic proteins. So it is part of the gene therapy. And these therapeutic proteins come from these gene that you deliver. And then vaccinate a person against an infection. If there is an infection and you vaccinate a person for that infection, that is also an objective of gene therapy. So only and the rest of the options as well, like replacing a missing or defective gene and repairing a non-functional gene. All of these are objectives of gene therapy, but it does not lead to cleaving of healthy genes which are already present to generate haploid cells or to cause any dysfunction at a cellular level. Okay, moving on to the next question. What property of the polymer causes proton sponge effect?
Secondary and tertiary amines.
Okay.
Negative surface charge.
Okay.
Option 2.
Okay. Anybody else would like to answer? What property of polymer causes proton sponge effect?
Second option, ma'am.
Okay. Okay. Most of you are correct. It would be presence of secondary and tertiary amines. So this is proton sponge effect is basically related to uh, particle escape from endosomal or lysosomal vacuoles or membranes. So particles are uptaken through endocytosis and they get localized in this endosomes or lysosomes. And to escape this, if you design the particles with secondary and tertiary amines, they basically will buffer the pH of these endosomes, which are mostly acidic, and that this will result in more proton influx through the endosomal proton pumps, which is followed by chloride ion transport and leads to osmotic swelling and bursting of these vesicles, which releases the particles inside this endosomes and lysosomes into the cytoplasm. So presence of secondary and tertiary amines would be the correct answer. Okay, we have another question here. Which of the following can be considered as an extracellular barrier for gene delivery?
Option 1 and option 3.
1 and 3. Okay.
Option 1 and 3.
Okay. Anybody else would like to answer?
Option 2 and 3.
Okay. So the correct answer would be option 1 and 3. Inhibition of transfection by mucus and degradation of DNA in plasma. So these are like extracellular barriers. So here gene delivery is basically you are either using some vectors or particles. If they get trapped by this mucus, it will prevent the transfection of them and uptake into cells, and then if delivered into the system, it can also get degraded in the plasma. Apart from that, there are other extracellular barriers as listed here, where inability to target DNA to specific organs and largely ineffective via the oral route except for immunization, and transfection inhibited by an uptake of DNA by reticuloendothelial system. Okay, moving on to the next question. So, if you are designing a cancer vaccine targeting new antigens, which are unique mutations in cancer cells, what is a key challenge associated with targeting new antigens for therapeutic purposes?
Option
Yes, which option? Yes, which option you think would be the correct answer?
option 3, ma'am.
Option 3. Okay. Others? What is a key challenge associated with targeting new antigens for therapeutic purposes? Anyone else? Okay, option 3 would be the correct answer here. So, can you please mute yourself if you are not going to say anything? Uh, Vishwas, okay? There is a lot of background noise coming. Can you please mute yourself?
Yes, ma'am.
Yeah, thank you. Okay, so new antigens are basically proteins that form in cancer cells due to mutations in the tumor's DNA. So the DNA, since it is associated with the tumor's DNA, it is very patient specific, and thus it is very difficult to identify due to the patient specific nature, of course, and it is very resource intensive and very time consuming. But uh, it can lead to very patient specific therapy and personalized medicine development. Okay, so the correct answer would be option 3. New antigens are difficult to identify due to their patient specific nature. Okay, moving on to the next question. Which of the following best describes the Broman effect in protein adsorption on biomaterial surfaces?
Option 2.
Okay. Anybody else? Okay, option 2 is the correct answer. So Roman effect is the where the initially adsorbed proteins are displaced by proteins with higher affinity over time. So if you introduce a biomaterial or any foreign material in presence of these cells and proteins, so if all of these are introduced into the system, first water will come in contact with your material and then followed by proteins and then cells. And these proteins, the nearby proteins, whatever is there, will start absorbing onto the surface first, and then more stable interactions is favored over time, which replaces this initial interaction of the proteins. So option 2 would be the correct answer. Initially adsorbed proteins are displaced by proteins with higher affinity over time. Okay, we have next question here. In biomaterials, what is the primary goal of pre-adsorbing proteins like fibronectin onto a biomaterial surface? Anybody? Okay. The correct answer here would be to enhance cell attachment to the biomaterial. This is the primary goal. So fibronectin is a protein of the extracellular matrix or ECM, and it binds to receptor proteins called integrins that are membrane bound proteins in several in all cells and in most of the cells, and these integrins bind to fibronectin. So if you want to increase cell attachment, you can use this fibronectin. uh, However, it can also lead to non-specific protein adsorption as well, but it does not prevent non-specific protein adsorption. So option 3 would be the correct answer, to enhance cell attachment to the biomaterial. Moving on to the next question. In cancer immunotherapy, why are dendritic cells used as antigen presenting cells for cancer vaccines?
Option 2.
Okay. Anybody else would like to answer? Yes, option 2 would be the correct answer. They are capable of activating both helper and cytotoxic T cells. Okay, another question. In nanoparticle based drug delivery, what is the primary role of surface pegylation? You have pegylation. Peg here is polyethylene glycol, which is another polymer, and it is most of the nanoparticles are coated with this peg molecules, which is also called surface pegylation. What is the primary role of this pegylation in nanoparticle based drug delivery?
Option 4.
Option 4. Okay. What about others? Anybody else would like to answer? In nanoparticle based drug delivery, what is the primary role of surface pegylation? Okay, so the correct answer here would be option C, to extend circulation time in the bloodstream. So when you attach this PEG molecules on or surface coat them on surface of nanoparticles, this basically prevents interaction with other proteins. You must have gone through the lectures where about brush conformation and mushroom conformation of these pegylation was taught. So mushroom conformation is basically like, so mushroom conformation of this peg molecule is basically like mushroom shaped peg coating on surface of your uh, nanoparticle or any other drug delivery vehicle, and this leave some space for interaction with the proteins, whereas if you have brush conformation, it it's kind of flowy, and as your proteins and other as your proteins come towards your nanoparticle, it gets these brushes prevent their interaction with your actual particle, and this kind of extends your retention time of these or circulation time of these nanoparticles in the system, and by preventing interaction with the proteins. Okay, so these were all the questions I had for today. Any doubt or any other thing you want to discuss? If not, we can close this session here, and I'll see you all next week.
Thank you, ma'am.
You're welcome. Do you have anything you want me to discuss again, or any doubt?